This section lays out the single constraint that the whole of immunity falls out of. The space of possible invaders is vast. It runs from a 30-nanometer poliovirus to a meter-long kidney worm. So the defense must generate recognition diversity that matches that vastness. The chapter's move is to reframe the immune system. It shifts it away from a search and destroy module and toward a recognition problem, one solved by two coupled layers. Fast and generic innate sits over slow and specific adaptive, and the whole thing is held in check by regulation rather than let run free. That reframe is the scaffold. The rest of the book hangs its death. There is an understated turn here. This network does not sit apart doing security work. It interleaves with the endocrine, nervous, and metabolic systems, tuning homeostasis. So balance, not destruction, is the organizing telos. This section reduces the whole chapter to a single primitive. Protection is memory. A body faces an unbounded adversary, from the roughly 30-nanometer poliovirus to a meter-long parasitic kidney worm. It cannot pre-store a defense for each one, so it learns invaders by exposure and remembers them. Thucydides already saw the shape in 430 BC, when plague survivors could nurse the sick unharmed. Jenner and Pasteur only operationalized the same observation, a safe rehearsal, cowpox or an attenuated culture, buys protection against the real thing. Everything later in the book, clonal selection, innate versus adaptive, primary versus secondary response, is mechanism for that single move, recognition matched to diversity, tuned by history. And the system is woven into the rest of the body's control. It interleaves with endocrine, nervous, and metabolic regulation, which makes immunity a form of homeostasis, not defense alone. This section explains the cells, organs, and microenvironments of the immune system as a single structural idea. One progenitor radiates into every blood and immune cell. The hematopoietic stem cell is the single root, and the whole system is the tree of its differentiation. The chapter's move is this. The immune response is a spatial program. Where a cell sits decides what it becomes and whom it meets. Primary organs, the bone marrow and the thymus, are where cells are made. Secondary organs, the spleen, the lymph nodes, and the mucosal sites, are where mature lymphocytes are staged to meet antigen. Blood and lymph knit them into one body. The microenvironment is the instruction itself, never mere scenery. Chapter 1, named The Players. This one hands them their addressing scheme, anatomy as the choreography that makes recognition happen in time and space. This section explains how Chapter 2 gives immunity a body. Chapter 1 said, what defense is. Here, defense becomes anatomy, and two structural facts carry the whole chapter. The first fact is convergence, then divergence. Every immune cell, from the red cell to the lymphocyte, descends from one hematopoietic stem cell. A single root fans out into the entire lineage tree. The second fact is that the system is a solution to a search problem. Antigen-specific lymphocytes are individually rare, so the body cannot rely on force. It relies on architecture. Primary organs build the cells. Secondary organs concentrate rare partners until they collide. Blood and lymph stitch the dispersed whole into one circuit. The generative primitive is that function is geometry. Immunity works because its cells are placed, not merely present. This section explains the single primitive that every immune cell reduces to. Every immune cell reduces to one primitive. A receptor binds its cognate ligand, and that binding is transduced into a response, divide, differentiate, move, or secrete. Recognition and response are one machine. A bond that fails to hold long enough to signal recognizes nothing. The chapter's compression is that adaptive antigen receptors, the B-cell receptor and the T-cell receptor, together with innate pattern recognition receptors and cytokine receptors, are three specializations of a single signaling logic. The same weak non-covalent bonds, made specific through multivalency and avidity, feed the same downstream pathways. The immune system did not invent recognition. It tuned the universal cell signaling loop against two hard constraints, a breathtaking antigen space and cells dispersed across the entire body. This part explains why molecular recognition is ordinary chemistry. A receptor holds its ligand by the same individually weak non-covalent bonds, each about 1 angstrom in reach, that any enzyme uses on its substrate. Specificity is only complementarity, summed over enough of those bonds to keep the two surfaces together long enough of those bonds to keep the two surfaces together long enough for a signal to pass. So the entire adaptive apparatus is generic. Cell signaling, bind, transduce, respond. It has two knobs turned hard. The first is the breathtaking diversity of receptors. The second is an affinity tunable from an enzyme-like dissociation, constant, or Kd, of 10 to the minus 5th molar, down to 10 to the minus 5th molar, down to 10 to the minus 12th molar, by mutation and selection across a response. Specificity is a number, a continuous quantity, rather than a discrete kind. Given a receptor's Kd and its residence time, you can already predict whether it signals. This section explains innate immunity, the fast arm of the immune system built on a bet evolution can make in advance. Evolution cannot anticipate a particular pathogen, but it can anticipate pathogen classes, the lipopolysaccharide of a gram. Negative wall, double-stranded viral RNA, unmethylated, KCOJ, every encounter. Innate immunity is the arm built on this bet. A germline-encoded set of pattern recognition receptors reading conserved molecular signatures, receptors reading conserved molecular signatures that self never wears. These signatures are called pathogen-associated molecular patterns. Identical every encounter. The step that makes this arm powerful is that the fast layer is simultaneously a classifier. Which receptors fire sorts the invader into a category, and that verdict is handed forward to the adaptive arm, which cannot begin until innate has spoken. Recognition is precompiled, not learned. The price is course resolution. The price. This section explains what innate immunity is for in the book's arc. Innate immunity recognizes the invariant rather than the individual. Its germline fixed receptors bind molecular patterns that are conserved across entire classes of pathogens. These conserved signatures are the pathogen-associated molecular patterns. Because the patterns are fixed in the germline, the response is ready before the enemy is ever identified. Immediacy is bought at the cost of resolution. The system distinguishes pathogen from self, never one pathogen from another. The chapter structures this as 3 ordered lines of defense. First, the physical and chemical barrier, then the cellular response, then the adaptive system. Each line is engaged only where the prior one is breached. Here is the move that makes the whole arc go through. Innate recognition does more than stall infection. It decides whether the slow, specific adaptive response fires at all. This system is ancient, shared with invertebrates. It is the substrate that adaptive immunity was later grafted onto. This section explains the complement system, and it opens with the single shape that everything else follows from. Complement is a cascade of latent enzymes, each one cleaved into activity by the one before it. Once you have that shape, the rest is consequence. 50. Odd serum zymogens sit inert until a pathogen surface trips the first of them. Cleavage begets cleavage, so one binding event amplifies into thousands of active fragments. 3 initiation pathways, the classical, the lectin, and the alternative, differ only in what trips them. All 3 funnel through a single node, the C3-C5 convertase, whose products then fan back out into opsonization, inflammation, and membrane punching. Ehrlich named the system for that last act, the thing that completes antibody. The chapter's move is to demote it. Lysis is only a sliver of what a self-amplifying, self-regulating proteolytic funnel actually does. This section explains the primitive behind the body's complement system. A cascade is the primitive, some 50 serum zymogens where each cleaved fragment activates the next, so a single binding event amplifies into a proteolytic avalanche. The avalanche converges on one pivot, the C3 and C5 convertases, from which every effector radiates. Opsonins tag, anaphylatoxins inflame, and the membrane attack complex punches holes. The surprise sits in the name. Ehrlich called it complement because it seemed to merely complete antibody. But the cascade is autonomous, and its specificity is negative. Recognition is promiscuous, the system runs by default, and self-survives only because host cells carry regulators that switch it off. So discrimination lives entirely in where activation is permitted. What gets recognized barely enters into it. Predict the rest, knock out a regulator, and complement turns on you. This section explains how a genome solves a storage problem. It cannot afford. A genome too small to store its outputs stops storing outputs and stores parts. The whole argument turns on one arithmetic embarrassment. Encoding each antibody separately needs about 10 times more DNA than the mouse genome holds, yet the repertoire exceeds 10 to the 7th power. The resolution is Tanegawa's, and it cost the one gene, one polypeptide dogma its life. Developing lymphocytes cut and splice their own DNA, assembling each receptor gene from a small library of interchangeable V, D, and J segments. A finite alphabet under combinatorial recombination generates an effectively unbounded space. The germline stores a generative grammar rather than a lexicon. From here you can predict the rest. Diversity will scale with how many ways the segments can be chosen and joined, which has nothing to do with genome size. This section explains how the immune system builds its antibody repertoire without storing it in the genome. The genome never stores the antibody repertoire. It stores the parts and a rule for shuffling them. A few hundred V, D, and J gene segments cut and rejoined in each developing lymphocyte combinatorially generate more distinct receptors than there are genes to encode them. The diversity is manufactured during development rather than inherited from the germ. This is Tanegawa's break with one gene, one polypeptide. The DNA of a B cell or a T cell is physically cleaved and recombined, so the mature receptor gene did not exist in the germline. The chapter's whole arc is this one move. A finite alphabet plus a stochastic assembler out covers any exhaustive lookup table. Now predict the corollary. Variety scales with the product of the segment counts, not their sum. This section explains how the immune system solves a visibility problem, how a T cell learns what is happening inside a cell it cannot enter. A T cell cannot see inside another cell, so evolution forced the cell to turn itself inside out. Every nucleated cell audits the publication and patrolling T cell of its own proteins and rivets the fragments to its surface, each one held in the groove of an MHC molecule. Surveillance becomes mandatory self-disclosure. The cell publishes what it is making, and patrolling T cells audit the publishes. Two channels split by provenance. Class 1 exports the cell's internal proteins, both self and viral, to killer T cells. Class 2 exports what a professional cell has swallowed to helper T cells. Here is the constraint that the whole system depends on. A T cell reads peptide plus MHC together, never free antigen. This makes recognition inseparable from the cell's own display vessel. This section explains how the immune system reads what is happening inside a cell without ever going in. T cells cannot see free antigen. They read only fragments held in the groove of a surface protein, the MHC molecule. That is the whole move of the chapter. To police the interior of every cell without entering it, the body forces each cell inspect, to continuously chop up its own internal proteins and post the peptides on its own surface. Intracellular state becomes externally auditable. Infection or malignancy leaks into a permanent surface census that patrolling T cells inspect. The generative primitive is surveillance by forced disclosure. And because these display molecules are hyperpolymorphic between individuals, the same census that reveals self also marks tissue as self versus foreign, which is why transplant rejection is what first named the locus. This section explains how the thymus builds a useful T cell repertoire. A useful repertoire is carved by controlled death measured against a self-reference. Progenitors enter the thymus, rearrange their receptor genes at random, then face two opposed verdicts on how tightly that receptor grips the body's own MHC bound to a self-peptide. The two verdicts point inward from either side, grip nothing, and the cell dies by neglect. That verdict is called positive selection. Grip too well, and the cell dies by deletion. That verdict is called negative selection. Only the intermediate band lives. The whole chapter is one primitive, development as a band-pass filter on affinity for self, carved by two death sentences pressing in from opposite edges. You can predict the rest from this. The surviving cell must see self weakly enough to ignore it and strongly enough to be anchored by it. That tension is the thymus's entire job. This section explains how the thymus builds its population of T cells. The thymus does not design T cells. It overproduces them blindly and then curates. Random DNA rearrangement gives each thymocyte a unique, untested receptor. Millions of specificities in all, most of them useless or dangerous. The shaping is done by selection rather than by synthesis. A cell survives only if its receptor binds self, MHC, weakly enough to be useful, yet not so strongly that it would turn on the body. Two thresholds bound a single survival window. Positive selection sets the floor. The receptor must engage the presentation frame at all. Negative selection sets the floor. 95% of thymocytes fall outside the band and die by apoptosis. The generative move is Darwinian, miniaturized. Diversity is cheap and random. Fitness is a two-sided affinity filter, and the repertoire is whatever clears it. This section explains the structural logic of B-cell development. A lymphocyte earns its existence by assembling the very instrument that will judge it. B. Cell development is a checkpoint, pipeline. Build part of a receptor, then let that part signal whether to proceed. The heavy chain is tested through the pre-BCR, the whole receptor through the BCR. Gate. Signal and product are one molecule. Construction and verification collapse into a single act. And the test, confirming that the receptor works, is also the test. Asking whether it binds self. Function and tolerance are screened by one apparatus. The rest predicts itself. A stage with no passing signal dies by default. Survival is licensed, never assumed. Diversity is generated blindly, then filtered. This is clonal selection, run inside the marrow. This section explains how B cells develop. The central idea is a pipeline in which the half-built receptor is its own pass or fail gate. Development from stem cell to mature B cell runs as a sequence of checkpoints rather than a fixed schedule. Each checkpoint tests whether the last immunoglobulin rearrangement produced a signaling competent receptor before it licenses the next round of division and the next rearrangement. Walk the sequence. When the heavy chain succeeds, the pre-B cell receptor fires, and that firing drives proliferation. Then the light chain rearranges, and the resulting immature immunoglobulin M is tested for self-reactivity. 2 problems. 1 machine. The same pipeline generates a vast combinatorial repertoire, yet purges whatever binds self. This is the T-cell primitive from Chapter 8. Diversify, then filter. Run here without a thymus. This section explains how a naive T-cell commits to a fate, differentiates into a helper subset, and finally persists as memory. A naive T-cell carries a receptor, but no fate. The antigen it binds selects which cell responds, while the context of that binding decides what the cell becomes. Activation is an AND gate. Signal 1 is antigen specificity. Signal 2 is costimulation, confirming danger, and both are required. So a self-antigen met without alarm yields tolerance, not attack. Past the gate, the ambient cytokine milieu steers the identical committed cell down one of several helper branches, each a distinct downstream instruction set. A fraction then settles into a low threshold, long-lived state. That persistence is the chapter's quiet surprise. Memory is hysteresis, the system holding its acquired configuration long after the stimulus that set it has decayed. This section explains how a naive T-cell gets its function assigned. A naive T-cell arrives with its specificity already fixed, 1 receptor, 1 antigen, but its function still blank. The move of the chapter is that the antigen-presenting cell writes that function in using 3 signals rather than 1. Signal 1 asks, "Is this your antigen?" Signal 2, costimulation, asks, "Is it genuinely dangerous?" Without it, the cell goes anergic, tolerant. Signal 3, the local cytokine flavor, asks, "What kind of danger?" and collapses the cell into one of a surprising fan of helper lineages: TH1, TH2, TH17, TH1, TH2, TH1, TH2, TH17, TFH. So response type is not something the lymphocyte carries in. It is read off the pathogen by the APC and imprinted at the moment of contact. Same cell. Context decides the fate. This section explains how B cells mount the adaptive response and the one idea that makes all of it predictable. A lymphocyte is chosen for already fitting its antigen rather than learning it. A B cell whose receptor binds gets activated. For protein antigens, this demands a second signal, help from a T cell. The activated cell then proliferates, and that single clone's descendants split into 2 fates. The first fate is plasma cells, pouring out antibody now. The second is memory cells, which wait, primed to answer faster later. Inside the germinal center, the binding that triggered activation becomes a fitness test. Receptors somatically hypermutate, and the higher affinity variants outcompete the rest. This is Darwinian evolution, compressed into days, run inside one lymph node, variation by mutation, differential survival by binding strength. Memory is simply the branch of that selection you keep. Once you see selection rather than instruction, the whole adaptive response predicts itself. This section explains how one immunological idea becomes physical structure. Burnett's clonal selection is one idea made flesh. The antigen does not instruct a B cell, it selects the one that already fits. What began as a 1957 postulate is turned here into anatomy. The germinal center is that mechanism, a bounded arena where a selected clone proliferates, deliberately mutates its own receptor, then competes for antigen and for T-cell help, the poorer binders dying. Variation, selection, amplification. Darwinian evolution run at somatic timescale, days long on one lineage. The output is twofold. First, plasma cells secreting antibody now honed to higher affinity. Second, memory cells held in reserve against the pathogen's return. The chapter's work is showing that adaptive immunity's founding abstraction is literally a Darwinian loop with a street address. This section explains how the immune system disposes of a pathogen once it has been recognized. One axis governs the whole chapter, where the pathogen lives. Antibody reaches only what floats free: the extracellular bacteria, the toxins, the virions in transit, coding them for neutralization, for phagocytosis, for complement. Anything that has slipped inside a host cell is invisible to antibody, so it must be handled through the cell itself. Cytotoxic T cells kill the infected cell outright, or T helper 1 activated cell outright, or T helper 1 activated macrophages digest what hides in their own vesicles. Recognition was the earlier chapter's problem. This one is disposal. And disposal partitions cleanly by accessibility. The elegance is predictive. You fix the effector arm from a single fact about the pathogen's address, never its identity. Humoral immunity for the reachable. Cell-mediated immunity for the concealed. This section explains the effector response, the stage where recognition finally becomes killing. Everything before this chapter built specificity. Nothing yet killed. The effector response is where recognition finally cashes out as clearance, and its generative move is decoupling. The molecule that finds the target is not the mechanism that destroys it. An antibody's two ends do two jobs. The variable Fab identifies. The constant FC dispatches, selecting which killer executes and where it reaches, complement, phagocyte, or NK cell. The same split runs the cell-mediated branch. The T-cell receptor recognizes. Perforin and granzymes execute. Read one effector mechanism and you can generate the rest. Each is a recognition event routed to a distinct execution module. The chapter's whole catalog is one pattern wearing different uniforms.